All right, so we are here to talk about some learning targets. The first few are going to be reviewing or kind of giving an overview of cellular respiration, and then we're going to move into fermentation. So these first ones are just going to be reviewing that respiration. So the first one says, I can explain the purpose of cellular respiration. Well, cellular respiration is a metabolic pathway. So metabolic just means it has to do with metabolism. And if you'll remember, metabolism is just all of the chemical reactions in your body. They can either be building up, which is anabolism, or breaking down, which is catabolism. And so when we talk about a metabolic pathway, we're just talking about a series of chemical reactions that are either breaking up or building down. And in this case, we are breaking down. So the purpose of cellular respiration is to break down glucose, this 6-carbon molecule, or some other fuel. We'll talk about that in one of the other videos. But to break down this fuel, this food, this energy source, which is glucose, into ATP. And so the goal is to make 34 to 38 ATP. In a perfect world, we would make 38, but it's not perfect. We lose energy here and there, and so it's not all converted to ATP. So very rarely do we get all 38 ATP, but that's, in a perfect world, that's what we would get. So the purpose of respiration is to produce or to convert the energy in glucose to ATP so that the cell can use it. And the reason we do that, why doesn't the cell just use the energy in glucose, is glucose is kind of like a $100 bill and ATP is like a dollar bill. And if you're going to buy a pack of gum, you don't really want to use a $100 bill because you know, what if the wind blew and it blew some of the bills away? I mean, that's kind of the analogy there. If you burst up your glucose all at one time, the cell is not going to be able to capture all that energy. Most of it's going to be lost as heat, and it's going to vaporize the cell. So if we can convert it to a smaller amount of energy, like ATP, then the cell can use that amount of energy. Not as much energy is lost. It's more efficient, and it doesn't, kill the cell, which is always a benefit. All right, so the purpose, again, break down glucose into to use that energy to make ATP. I can list the processes that make up aerobic cellular respiration. So these are the 4 processes. Glycolysis is first. It makes pyruvate, which goes into pyruvate oxidation, which obviously is where pyruvate is oxidized, meaning electrons are removed. That leaves us with acetyl-CoA. Acetyl-CoA goes into the citric acid cycle, where even more electrons are removed. So we're going to oxidize that acetyl-CoA in other forms, that citric acid, that 5-carbon. And then once we've oxidized all those, we've gotten all of those electrons, because remember, oxidize means to lose electrons. Then we take all of those electrons and we send them down to oxphos. And we'll talk about that in just a second. So oxidative phosphorylation is going to be the main part that converts that energy into ATP. There's a few ATP made here and a few made here, but the vast majority are made down here. I can name the locations of each of the 4 processes that make up aerobic cellular respiration. Glycolysis happens in the cytosol. Okay, we didn't talk about this, or depending on the year, we didn't talk about it this year. Pyruvate oxidation, I believe, occurs in the intermembrane space. It is in the, it is inside the mitochondria, and pictures and diagrams I've seen and things I've read seem to indicate that's where it's happening. And then the citric acid cycle is occurring in the matrix, which, if you'll remember, the matrix, let's draw a quick little, over here in our extra space, little mitochondria. Okay, so glycolysis is happening out here. Pyruvate oxidation is happening in here. The citric acid cycle is happening in there. And then oxidative phosphorylation is going to happen on the inner membrane. Okay. So that's our next I can statement. And then the last one is I can explain the movements of electrons and therefore energy in respiration. So the whole idea, or the whole point behind respiration, is to release the energy from glucose. And the way that we do that, for the most part, is through electrons. So electrons are released and they're carrying energy with them. And so when we talk through this, you'll hear me say, okay, anytime you break a bond, energy is released in the form of, and y'all say, electrons. Because that is what's happening. When this glucose is broken in half into these 2 3-carbons, electrons are released and they're carrying a lot of energy. And that energy and those electrons goes to NAD+ to reduce it to NADH. It's being reduced because it's gaining electrons. Okay, then that is going to go down to oxidative phosphorylation. Over here, Pyruvate, we're going to break off another carbon. So again, anytime you break a bond, energy is released in the form of electrons. It goes to this NAD plus to reduce it to NADH. There are actually two of them because this is happening twice. And they're going to go down to oxphos. Then over here in the citric acid cycle, we broke off a carbon, released electrons and energy, broke off another carbon, released electrons and energy. This says high. You can't really read that. Let me zoom in a little bit, maybe. Still can't really read it. It says high E for high energy and low E for low energy. And so what we do is we have this high energy molecule, and we'll make y'all seasick if I keep doing that. We have this high energy molecule, and we are breaking the bonds and replacing them with low energy bonds. And when we do that, we release some electrons to reduce NAD plus and FAD plus plus, and we also release a little bit of energy to make ATP. This is also happening twice. So we see 1, 2, 3 NADHs, but it's happening twice. So we're going to see 6 NADHs coming from here and 2 FADH2s. Okay, so it says, I can explain the movement of electrons. At this point, we're seeing electrons move from glucose or pyruvate, which is, you know, what's the word I'm looking for? Product, I guess you would say, a subsidiary of glucose. It's still glucose. It's just broken in half. And then acetyl-CoA. These are the two carbons from acetyl-CoA that get broken off. And acetyl-CoA comes from pyruvate, which comes from glucose. So when we talk about these carbons all coming off and these electrons all being released, all of these electrons are coming from glucose. So the electrons are moving from glucose to NADH or FADH2. Okay, mostly NADH, right? There's 10 of these and only 2 of these. So vast majority is going to that. And then what's going to happen here is we are going to see NADH drop off those electrons and FADH2 drop off its electrons into this pathway. Okay, and they're going to go through a series of redox reactions as they go through that electron transport chain. So what we're seeing now is the electrons have moved from glucose to NADH and FADH2 to the electron transport chain. You could also call those proton pumps, but it's actually going to these molecules. And then if you'll remember, at the end, the electrons give all of their energy to these molecules. They use that energy to pump H's up, right, into that high concentration space. And then when the electrons get tired, they are given to, let me see what's a good color for me to use down here, O2, oxygen. So oxygen is the final electron acceptor. So they are given to oxygen, and then oxygen grabs a few of these H's and makes water. I guess I'll put oxygen and not water. So this is the path of electrons. Okay, if we were to look at the path of energy, it is mostly the same, but it is a little bit different. Okay, so it starts in glucose. Let me do it in a different color just so it's not confusing. So the path of energy is from glucose. Okay, it's going to be in electrons to start with, so NADH and FADH2. From there to the electron transport chain. They're going to use that energy to create a stronger concentration gradient. So it's going to go into the potential energy, capital E is the abbreviation for energy, of concentration gradient. That's a lot to write. So potential energy of concentration gradient, which is then used to, that potential energy is relieved as the H's fall through here. Okay, that causes that to turn. So that's mechanical energy because it's literally moving, kinetic energy you could also say. And then that is given to ADP to phosphorylate it to ATP. And I didn't really show that. Let's see if I can add that in there. So ATP from ADP. Okay, so if you're looking at electrons, energy, they start off the same, but when it gets to the ETC, the electrons have done their job. They give their energy to the electron transport chain, so they have no more energy and they just go hang out with water, or excuse me, with oxygen to make water, but the energy is not done. The energy is used to create this high concentration up here, which then falls through, causing this to turn, and that provides the energy needed to form that bond between the second and third phosphate to make ATP. So that is how you can explain the movement of electrons and energy in cellular respiration. I hope that helps. If you still have questions, let me know.